Yeah, welcome. Thank you for coming to my talk where I will tell you about how studying the evolutionary dynamics of microRNAs can help us pinpoint innovations in the regulatory network of vertebrates. So let's all get on the same page about microRNAs. So microRNAs are short regulators of gene expression, and they bind through mRNAs by binding with a 6 to 8 nucleotide long motif, which is called the seed sequence. And the issue we're tackling here is that the annotation of these microRNA seed sequences of the microRNAs come from transcriptomic data, which in turn is specific to the conditions in which the sample was taken. So here, for example, we have in human, we have a large catalog because we have lots of samples that come from different tissues like brain or bone or the intestines. So in these model organisms, the microRNA catalogs are very complete, whereas in non-model organisms, the catalogs are only fragmented, if available at all. So what we set out to do is to take the catalogs from these model organisms and shift the annotation to non-model organisms. And the method with which you do this is by performing an ortholog search. So an ortholog search of microRNAs is challenging for multiple reasons. First, the mature microRNA transcript is very short, and also the genes can be located in repeat-rich regions. And both of these factors make sequence similarity searches like BLAST-N lose specificity. And the solution that we came up with is that, first of all, well, we use the precursor microRNA sequence, which has at least 60 nucleotides. And really the key factor here is that we first shrink the search space by using the conservation of gene order. And the way we do this is that we first pick one reference species that has which is a model organism with a complete microRNA annotation, or mostly complete. And then we first identify a few relatively closely related organisms, here, for example, different primates. And in this small set, we gather training data by first looking for each microRNA at the protein coding genes immediately downstream and upstream of the microRNA. And then we use these protein coding genes as synthetic anchors to identify microsynthetic region in our 3 core species here. And with these high confidence, positionally informed orthologs, we can then use those as training data for training a covariance model. And a covariance model you can think of as a profile hidden Markov model, which can also model the additional constraints of secondary structure in RNA. So now in the next step, we have our model. And now we take this model to perform a model-based ortholog search, which doesn't take gene conservation gene order conservation into account. It just works from the model. And this gives us these presence-absence patterns of phylogenetic profiles of our microRNA of interest across large collections of taxa. And if we now compare our inferred orthologs with a database, with MirGeneDB, we treat this as a gold standard database because the data in there comes from experimental data, which is manually created. And if we compare our inferred orthologs with this, we get an accuracy of 93% and a sensitivity of 97%. So not too bad. So what can we now do with these catalogs of microRNAs in non-model organisms? So one thing you can do is that with the number of co-orthologs that we detect with our method, we can actually very nicely track whole genome duplication events, for example, the 3RD event in the teleostei, but also lesser known whole genome duplications like the one in Xenopus levis. So if you're interested in tracking whole genome duplications, microRNAs might be a good marker for that. Next, we now have the possibility to use pre-microRNA sequence as a phylogenetic marker for tree reconstruction. We're not the first ones to use pre-microRNAs as phylogenetic markers, but now we have the possibility to use them in basically any taxon set, and we're not restricted to these smaller collections anymore. And the reason you might want to do this, the reason you might want to use microRNAs as phylogenetic markers, is that we actually find evidence that this secondary structure, this hairpin, gives additional constraints to the sequence that help to resolve quickly speciation events in quick succession. So the keyword here is incomplete lineage sorting. So if that's something you're faced with, microRNAs might be a good way to go. So now let me take you back to these phylogenetic profiles where we have the presence absence of microRNAs. We have here in the rows 267 microRNA families in human, in 556 vertebrates here in the rows. And from these profiles, you can see a couple of things. First of all, you have these phylostrata of where microRNAs have evolved and have different ages. We have very old microRNAs over here, younger microRNAs over here. And you can also see that here we have more orange dots. So that means that the seed sequence is not conserved. So these younger microRNAs, seed sequence seems to be more fluctuant. What else you can see from this, I hope, is that you can actually, with these larger collections of phylogenetic profiles, you can make more easily the difference between spurious absences, like the small dots here, and large lineage-specific evolutionary events where the absence of microRNAs occur on a whole lineage. And since these kinds of events, these gaps in the profile are so rare, we took one of them and characterized this event of loss of 60 microRNA families in rodents in a little bit more detail. And the few things that we learned for this, I just want to go quickly over it. First thing we checked is that each and every of these microRNAs, these 16 families, have unique seed sets. So there's no other rodent-specific microRNAs that potentially have taken over the role of these microRNAs, which could have prompted their loss. Also, we overexpressed two of those microRNAs in human and mouse cells, and we see that there's actually an overlap in between the two model systems. So there seems to be parts of the ancestral regulatory network still intact, which leads us to believe that there was an ancient regulatory network which was impacted by the loss of these microRNAs. And last, we then checked if maybe the rodents have first lost the targets of the microRNAs, which then allowed them to lose the microRNAs afterwards. But if we look at the profiles of the protein coding genes, we can actually see that the putative targets of these microRNAs in rodents, they are all conserved. But what we can also see that the genes that are lost in the rodents, so on the same lineages here, that these genes are rich in transcription factors. So looking at these microRNA profiles really allowed us to track a large structure rearrangement of the regulatory network in rodents, which appears both on the transcriptional level of the transcription factors and the post-transcriptional level of the microRNAs. And with that, I would like to thank you for your attention. You have a QR code to the publication over there. And I would like to thank all my group and the EvolCompGen COSI for inviting me and funding my travel. Thank you.
Hello, yes. Yeah, perfect. Just about where they are located in the genome, this microRNA, are they next to genes, next to telomeres? Like, where are they scattered across, or are they together?
They are pretty scattered across. So most of the human ones are intergenic, but there are also a subset, I don't know the percentage off the top of my head, which are in introns or even inside of coding regions.
Okay, yeah. And so they have their own regulation, like transcription,
yeah.
Or do they hijack the mRNA transcription?
Yes, so both. So there are microRNAs, to my knowledge, with their own promoters, and there are others which are processed from introns.
Okay. Okay. Yeah. Thanks a lot.
Thanks.
We have a question from online. Priya says, "Hey, amazing talk. How do you select the reference microRNA? Are there any properties that the reference one should have?
So for the reference microRNA, what we did is just take all that was available in the MirGeneDB database. So anything that is a high confidence annotation of a microRNA, there's not really any limit of what you can use. Thanks.
I have a question about the loss in rodents. I mean, the fact that it's consistently across all the rodents means the loss happened already in the common ancestor,
Yes.
likely. I mean, these non-coding sequences, they evolve quite rapidly. I mean, do you have any sense how come the target is still conserved?
Yeah. Good question. So so we looked at the predicted target sites for these for the mRNAs that are targets, and for the mRNAs that still have a response, the target sites are actually older than the microRNA. So it seems to be that there's some other constraining factor that keeps the target site alive, which basically allows us to detect it now. Because all the other ones, they would be degraded, right?
Thank you.
Thanks.